We have just had an introduction to the nervous system and the neuroscience, and before we continue, it is important to learn on how the nervous system has developed. So in today's session, we're going to discuss the embryology of the nervous system. What's up, ladies and gentlemen? Welcome back to Neurosketch with Dr. Kenny. But if this is the first time you're seeing this channel, don't forget to subscribe. Today we're going to discuss about the embryology of the nervous system, the development of the nervous system. But as always, I like to go back to square one. We said the nervous system can be divided either functionally or anatomically. And in anatomical division, anatomical classification, the nervous system can be divided into the central nervous system and the peripheral nervous system. And remember, the central nervous system is made up of brain and the spinal cord, and everything else is included under peripheral nervous system. This includes the nerves, both cranial and spinal nerves, the ganglia, and the sensory receptors. They are all falling under the peripheral nervous system. But in functional classification, the nervous system is divided into sensory system and the motor system. But the motor system is further subdivided into somatic nervous system and autonomic nervous system. Again, discussed about tissues, and we said the tissue is formed by a combination of cells. Now, the nervous tissue is formed by the two main types of cells. We have the nerve cells, which are the main cells of the nervous system. They are also known as neurons, but they have their supportive cells, which are called the glial cells, also known as the neuroglials. These are the two main types of cells that are making the nervous tissue. Now, let's go to our topic of discussion today, which is the development of the nervous system. And this is going to be easy because I've put this objective for you here. I mean, once you know these objectives, you are good to go. You are good to go to exam of the embryology of the nervous system. And it's easy. You just need to know when does it start, where does it happen, how does it happen, what are the faults, like what happens if something goes wrong, and what are the derivatives, because this is the development. So what do you want to achieve by the end of this process? And fun enough, I have included the answers for you, so your life is definitely easy. So when does it start? It usually starts from the 3rd week of conception, from the ectoderm. And how? By the process known as neurulation. But the neurulation is mainly for the central nervous system, which is the point of focus, central nervous system. And what are the faults that can happen in the development of the central nervous system? You have neurotube defects, for example, the anencephaly in the spinal bifida, but you also have the disorders like hydrocephalus. And what are the derivatives? What do you want to achieve? The central nervous system will give rise to brain and the spinal cord. And at the part of the brain, you have the forebrain, diencephalon, brainstem, and cerebellum. These are the main parts of the brain. And you also have the spinal cord. And we're going to spend a lot of time emphasizing on how does it happen, but let's start with where does it happen. As we have said, the nervous system is coming from the ectoderm. Remember the 3 germ layers, the ectoderm, mesoderm, and endoderm. Now the nervous system is coming from the ectoderm. So what is happening here is the ectoderm will undergo differentiation to form the surface ectoderm and the neuroectoderm. And as you can guess, the neuroectoderm is the one that will give the nervous system. And this process of differentiating from the ectoderm to the neuroectoderm is known as the neuroinduction. What is neuroinduction? The neuroinduction is the process of differentiation of the ectoderm to form the neuroectoderm under the influence of signal molecules that are coming from the notochord. Remember, notochord itself is not a part of ectoderm but mesoderm. Notochord is coming from the mesoderm, but it releases molecules that are going to induce the differentiation of the ectoderm into the neuroectoderm. And then from the neuroectoderm, we form things like neuroplate that are going to form the nervous system. Let me show you here. So this initially was the ectoderm, and the ectoderm underwent the differentiation under the influence of the molecules that are coming from the notochord to form the neuroectoderm. And the neuroectoderm will further thicken and differentiate to form the neuroplate. So this is the neuroplate, which is actually formed from the thickening and the differentiation of the neuroectoderm. How was the neuroectoderm formed? It was formed by the differentiation of the ectoderm under the influence of molecules of the notochord. So this is the neuroplate that we have here. And remind you that this is the neuroplate, this is the ectoderm, and between the neuroplate and the ectoderm we have this border, and which is called the neuroplate border. Next in the process is the invagination of the neuroplate to form the neurogroove. You know, the neuroplate folds upon itself to form the neurogroove. Here the neuroplate folds upon itself, invaginates to form the neurogroove here. So let me show you what happens is So you had a plate like this, and this is called the neural plate, and the neural plate has got neural plate borders that's different, that separates them from the ectoderm. So what happens is you have the invagination, folding of the neural plate to form a neural groove. A groove is a furrow, okay? And pay much attention to the borders, because the neural plate borders will approach one another, and they forming this the neural groove, and this is the structure that you are seeing here. Okay, so the neural plate border will the cells will differentiate to form the neural crest. We're going to talk about neural crest a little bit later, and also have this invagination, the points of folding, which I call the neural fold, and you have neural groove. So you have the neural groove, neural crest, and the neural fold. Next in the process, there's the formation of the neural tube. Now the edges of the neural groove will come together to fuse to form a tube, and that tube is called the neural tube. That is the neural tube. This one, let me show you by this one. So this is the neural tube here, and this tube is formed by the fusion of the edges of the neural groove. Okay, this is the tube, and the neural tube is very important because the neural tube is the one that gives the derivatives of the central nervous system. If at all you have the structures of the central nervous system, then they are coming from the neural tube. What are these structures? These are the brain, spinal cord, and the preganglionic autonomic fibers. All of these are coming from the neural tube. Then I want to orient you to the structures that are adjacent to the neural tube. This is the neural tube, and on the other side of the neural tube you have somites. You can remember somites are the one that will give rise to the bones of the exoskeleton, all the way from the cranium, the ribs, the vertebral column, and bones of the exoskeleton are coming from somites. And anterior to the neural tube you have the notochord, and the notochord will later degenerate, will later form the nucleus pulposus, which is the part of the intervertebral disc. The intervertebral disc, that the central part, which is the nucleus pulposus, that is the remnant of the notochord. Okay, this picture here summarizes the whole process all the way from the neural plate to the neural tube. Yes, this is the neural tube here, and this neural tube will later form the central canal of the spinal cord and the ventricles of the brain. The ventricles of the brain are coming from the neural tube. The central canal are coming from the remnant of this neural tube. And also, yes, so this is the notochord, and this is the somites on either side of the neural tube. You have the somites, and then this is the notochord. Okay, going back to the neural crest, I told you we talk about neural crest, and the cells of the neural crest will migrate throughout the embryo, and they will give rise into a large range of cell types. So it will form parts of the peripheral nervous system and the autonomic nervous system, and if you want to be specific, it will form things like melanocytes, the one that forms melanin. You have so cells of the sensory neurons, which are the afferent neurons, are coming from the neural crest, and also the autonomic ganglion neurons, or the postganglionic neurons of the autonomic system, they are coming from the neural crest, and this is how neural cresting contribute to the formation of the nervous system. Okay, let's go back to the neural tube because we didn't finish discussing about it. The neural tube so formed will grow in two directions, into the cranial direction and the caudal direction. And let me show you by here. Okay, so this is the neural tube that was formed, and this tube will undergo growth both cranially and distally. The cranial portion will form the brain, and the distal, the caudal portion, will form the spinal cord here. This is what I mean, like this cranial part will form the brain, and the caudal part will form the spinal cord. And I want you to notice this is the tube, and because the tube, it has got opening. Okay, it has got the proximal opening and the distal opening. This opening here is called the cranial opening, or the cranial neural pore, or the anterior neural pore, and this downstairs here is called the posterior neural pore, also called the caudal neural pore. So the anterior neural pore, also called the cranial neural pore, the posterior neural pore, also known as the caudal neural pore. What's so important about this opening is the day of closure. The anterior neural pore closes at the day 25 of conception. From conception to the closure, it takes 25 days. But the posterior neural pore, it closes 2 to 3 days after the closure of the anterior neural pore. You have the closure of anterior neural pore, and then the posterior neural pore stays open for 2 to 3 days to allow exchange of material between the neural tube and the amniotic fluid, the fluid adjacent. So it later closes at day 28 of the conception to completely close the neural tube. So the neural tube is closed. And if the neural tube does not close at these days, we have faults, and these faults we call them the neural tube defects. For example, when the anterior neural pore fails to close, we have a condition which is called the anencephaly. And anencephaly basically means absence of brain. This is a fatal condition because it is not compatible with life. Because if you lack brain, then you are not going to survive. And so it is a fatal condition. If the baby is born without brain, is as considered as dead already, because it's not compatible with life. And this occurs because of failure of closure of the anterior neural pore at day 25 of conception. Very, very important, and this will appear in your MCQs exam questions. Then to the posterior neural pore, if it doesn't close at day 28 of conception, it will result in the condition which is called spinal bifida or spinal bifida. And this is the defect in the vertebral column that is resulting to exposure of the content of the central nervous system. It can include meninges, or it can also sometimes include the spinal cord. And I have a separate discussion about these neural tube defects, and I have made a separate video discussing about this. To keep this discussion short, I have not included the details of the neural tube defects, but you can always find a video, a separate video talking about the neural tube defects. And again, I have not included also the discussion on the development of the spinal cord, and this will also come as a separate session. And here I am concentrating on the development of the brain to make this short session even more clear. Okay, so the neural tube that is formed, we've already seen how the neural tube is closed. Like we have anterior neural pore and posterior neural pore. They are closed at different days, day 25, day 28. The anterior neural pore, if it doesn't close, is that into the anencephaly, and the posterior neural pore, if it doesn't close, is that into spinal bifida. The details about this have the separate discussion. As we have said, the neural tube has got two ends, the cranial end and the caudal end, and the brain, we have said the brain is coming from the cranial end of the neural tube. So what happens is we have two constrictions of the cranial end of the neural tube. This is constriction number 1. This is constriction number 1. This is constriction number 2. As a result, it divides the cranial end of the neural tube into 3 primary vesicles. This is vesicle number 1, number 2, number 3. We have 3 primary vesicles that are coming from the cranial end of the neural tube due to 2 constrictions that are forming these 3 primary vesicles. Let me use this picture here. So this is constriction number 1, this is number 2, this is number 3. This is the vesicles, the constrictions here. This is constriction number 1 and this is constriction number 2. The result is 3 vesicles, number 1, number 2, and number 3. These vesicles are called number 1 the prosencephalon, number 2 the mesencephalon, and number 3 is the rhombencephalon. Okay, I want to give you a trick here. A trick is the word encephalon means brain. So the pros means the fore anterior. This is the forebrain. Yeah, the prosencephalon is the future forebrain, and the mes means mid. So the mesencephalon is the future midbrain, and rhombencephalon, this is the future hindbrain. This is easy. It's going to be easy. These are the 3 primary vesicles. But we're going to move from 3 primary vesicles into the 5 secondary vesicles. This is what happens. Like the prosencephalon will further subdivide to form the telencephalon and the diencephalon, and the mesencephalon will continue as the mesencephalon, and on the other hand, rhombencephalon will further subdivide to form the metencephalon and the myelencephalon. And mind you, I told you this is the future forebrain, this is the future midbrain, and this is the future hindbrain. Another views, this is just a different view, like we are moving from the primary brain vesicles to the secondary brain vesicles. We had the prosencephalon, mesencephalon, and the rhombencephalon, and we are going to form the prosencephalon will give the telencephalon, the diencephalon, mesencephalon will continue as the mesencephalon, and the metencephalon, the rhombencephalon will further give the metencephalon and the myelencephalon. These are called the secondary brain vesicles, and this is the spinal cord. Remember, we're talking about the brain, and this spinal cord is developing from the caudal end of the neural tube, but here we are in the proximal or the cranial end of the neural tube. One more time, we've said we have three primary brain vesicles, which are the prosencephalon, mesencephalon, and the lombencephalon. And we've said the prosencephalon will further subdivide to form the telencephalon and the diencephalon, but the mesencephalon continues. The mesencephalon does not divide, and the lombencephalon will divide to form the metencephalon and the myelencephalon. And this is what we've just discussed. Now, because these are vesicles, these are the five secondary brain vesicles, but because they are vesicles, that means they've got, let me show you, that means they've got a wall. They are vesicles, means they have a wall outside, and then inside they have a cavity. Okay, they have a wall and a cavity, and I want to show you the derivatives, like what we have from these vesicles. So from these vesicles, the walls of the vesicles will form these structures here, and the cavities within the vesicles, we are going to see the cavities here. So for example, the wall of the telencephalon will form the cerebrum, and I've told you the prosencephalon is the future forebrain. So the future forebrain contains the telencephalon and diencephalon, but the wall of the telencephalon is what forms the cerebrum, the major part of the brain. And what about the wall of the diencephalon? This is the one that gives the hypothalamus, epithalamus, and thalamus. Okay, so we have thalamus, epithalamus, and hypothalamus. Where do they come from? They are coming from the encephalon. Is the encephalon from the forebrain or midbrain? It is from the forebrain. Okay, but this is the wall, the outside. The wall of the diencephalon forms the thalamus, epi, and hypothalamus. The wall of the mesencephalon, as I've told you, it gives you midbrain. And what about the wall of metencephalon? Take care here, because the metencephalon will give to pons and cerebellum, not medulla. Pons and cerebellum. Metencephalon, the wall of the metencephalon will give rise to the pons and cerebellum, not medulla. And on the other hand, the myelencephalon, which is continuous the spinal cord, will give rise into the medulla or medulla oblongata. But these are just walls, as I've told you. Let me show you one more time. We've said these are vesicles, and the vesicles have got walls and the cavity inside. Now, for example, the wall of the telencephalon will give rise into the cerebrum, and the wall of the diencephalon will give rise into the epithalamus, thalamus, and hypothalamus. The wall of the mesencephalon will give rise to the midbrain, and the wall of metencephalon will give rise into the pons and cerebellum. And lastly, the wall of myencephalon will give rise into the medulla or the medulla oblongata. And don't forget, these are just walls. Inside they have cavities, and these cavities, they are forming the so-called the ventricles of the brain, chambers of the brain that are called the ventricles. And for example, in the telencephalon, you have two ventricles. We call them the lateral ventricles, left and right ventricle. And the reason we have two ventricles here is because we have two cerebral hemispheres. We have right cerebral hemisphere and the left cerebral hemisphere. Within each, there is the ventricle that is called the lateral ventricle. We have two lateral ventricles, and then we have the cavity within the encephalon forms the third ventricle. A cavity within the mesencephalon forms the so-called the aqueduct of midbrain, also known as the aqueduct of Sylvius, also known as the cerebral aqueduct. So the cerebral aqueduct of Sylvius, or aqueduct of the midbrain, this is found within the midbrain. You can never forget. And lastly, we are metencephalon and the mesencephalon. They all contribute to the so-called the fourth ventricle, whereby the upper part of the fourth ventricle is formed by the cavity of the metencephalon, and the cavity of the myelencephalon contribute to the lower part of the fourth ventricle. So in summary, we have, if you have noticed, we have four ventricles of the brain. We have left and right ventricle, which means two ventricles, and then we have the third ventricle, and then we have the fourth ventricle. This aqueduct is just a pathway. It's not a ventricle, but it's a cavity. Yes. Where is it found? It is found within the midbrain. This is very important. We're going to discuss later. We'll have a separate discussion on either part, but this is just a development. This is what happens until we form these parts of the brain. So this is summary, to summarize what I've been talking about here. short and conclude, let's see the major parts of the brain. So the adult brain consists of the cerebrum, the diencephalon, midbrain, pons, medulla, and cerebellum. But I want to make something very clear here, that midbrain, pons, and medulla together they are forming a continuous stalk, and this stalk is known as the brainstem. So as a result, the human brain has got four main parts now. Yeah, these parts are the cerebrum, diencephalon, brainstem, and cerebellum. And remember, the cerebrum and diencephalon are coming from the forebrain, midbrain is coming from the midbrain, and the hindbrain gives the pons, medulla, and cerebellum. And these are the derivatives. These are the derivatives of the development of the brain. So going back to where we started, we said you should know when does it start? Third week. Where? Ectoderm. How? Neurulation. But neurulation is specifically for the central nervous system. Okay, brain and the spinal cord. But the faults that we've seen, the neurotube defects like the anencephalus, spinal bifida, but the hydrocephalus is separate. We have a separate discussion about it. And also these neurotube defects, we have a separate discussion. We've discussed them somewhere else. And the derivatives we've just seen, we have the the brain and the spinal cord, but we haven't discussed about the spinal cord. So the derivatives of the cranial end of the neurotube is the forebrain, diencephalon, brainstem, and cerebellum. And this is what I had for you. So if you have enjoyed this discussion, don't forget to like, share with your friends, but subscribe to keep in touch, and see you real soon. Ciao.